So the third type of data that is generated and we will import in the R program, it will be from the Illumina company. So Illumina data comes in a variety of formats, and here we will discuss only one or two types of Illumina data. And the technology of Illumina is somewhat different from the Agilent and the Affymetrix. Because in the Illumina technology, small glass beads are used instead of glass slide on which the probes are attached. So here is not the time to differentiate between the procedures of the Illumina and the Agilent and Affymetrix, but it's just to remember that Illumina company, in the procedure they use small glass beads. So for the Illumina data, we will discuss only the one of the important variety of their data is called the bead summary data, and we can customize the format of this bead summary data. And one type of bead summary data is Bead Studio version 1 data, and this Bead Studio version 1 data uses the target IDs for the various types of genes for each gene, and these target IDs, they are used as identifiers for each of the gene. And the second type is Bead Studio version 3, and it uses the probe IDs, and for which the intensities of the probes is analyzed by these type of chips. And there are two types of data can be analyzed for the Illumina data, and these two are the bead array and Illumi. And we can use the command that is present in the R program is the read bead summary data, for which we can use the data that is present in the different folders, and we can use this command to analyze the Illumina data. Here we can see it's a long coding for Illumina. We can use this read bead summary data command to read either the probe IDs or target IDs. I have told you that the target ID can be used as identifier, and probe ID is used for, means different probes are used for different genes to analyze the expression of the genes. And then we can use the term skip is equal to 7. We will see what is this. So expression set Illumina, the assay data has 24,350 features, total number of 8 samples. So element names, they are present. So it's a long information that is present in this Illumina data, and this data can be imported into the R program by using this command, read bead summary data. So we can see the features of this data by reading all of these features that is present. So by using this command, we can import the Illumina data also in the R program. So we have used the read bead summary data command to import the Illumina data into the R program. So because this data is somewhat different from the Agilent and the Affymetrix data, so we have to use the column probe IDs to identify the different types of different sets of data present in the Illumina. So data, the command is like this: data read bead summary, probe profile. We have used probe IDs, and probe ID is separated into different. We have to skip the 0 in the previous. We have to skip 7, that are now in the object data that are expression set Illumina, we can check that everything is okay, then we can analyze this data. So by importing the Affymetrix, Agilent and Illumina data, we can now analyze these data sets by using the R program.